Hi everyone, this is Dharani Jagadisan, Assistant Professor of Biochemistry, A.D.M. College for Women, Autonomous, Nagapattinam. Today we will see the topic is Introduction of Immunology. Immunology is the study of host defense mechanism. Immunity is the ability of host to protect itself against pathogenic foreign organism. The immune system compares the tissue, cell and molecules which amount the immune response. This is the types of immune system. First type is natural. It is otherwise called as innate immune system. Skin, tears, saliva, mucus, acids, etc. Properly of all living things characterized. Adaptive immune response. This is otherwise called as accurate immune system. Specialized cell, cytokinase, antibodies. Specialized as memory. Specialized mucosal lymphoid tissue. This is the two major kinds of defense. One side is accurate immunity, then other side is innate immunity. Accurate immunity contains two types, lymphoid stem cell, then otherwise innate immunity system contains mucoid stem cell. Accurate system contains divided two types, one B cells and T cells. Then innate immunity contains basophils, eosinophils, neutrophils, mucocytes, platelets, erythrocytes. This is the immunogenicity and antigenicity. Immunogenicity. It is the capacity to induce an immune response by foreign, complex and high molecular weight compounds, mostly protein. Antigenicity. It is the ability to bind to Ig. Ig calls immunoglobulins or immune cells. And immune response may not rise. All immunogens are antigens, but all age need not necessary by immunogens. Haptens. Haptens carry translate. This is the innate immune system. A reliable means of protecting the host in the first intent agent many extracellular organisms. It is a property of every living organism. Unable to deal with an intracellular organism, etc., protozoa, virus and certain bacteria are not killed. Non-specific as no memory involved. This is also the innate immune system. System is the naturals or native immune system. Rapidly mobilized first line of defense. Not depends on periods exposure to the foreign innover. May not be to prevent foreign material in the hearts. This is the component of the innate immune response. Psychochemical barriers, molecules normally present in the body fluid. Example, lysosome, complement and anti-protein. Pathogenic and cytotoxic cells such as neutrophil, macrophages, natural killer cells. Natural killer cell is otherwise called as NK cells. This is the biological and chemical physical actions of immune system. Most of threat neutralization by the innate immune system. First of all, we see respiratory system, also known as oxidative rust. Membrane bond NADPH oxidized in combination with other enzymes produced. Superoxide anion, hypochlorous acid, hydrogen peroxide, chloroamines. This is the accurate immune system. Antigen specificity is the important of the accurate immune system. Antigen specificity is the single most important aspect of the accurate immune system. Each alone of the lymphoid cells responds only to the single antigen. T cells dies with suffered bond produced antigen. B cells dies with soluble extracellular native antigen. This is the humoral and cell mediated immunity. Specific immunity is termed humoral when antibodies are involved in removing the antigen. It is termed cell mediated when T cells and or the charges in the pathogen clearance. Active and passive immunity. It is the term of immunity because the heart does respond actively to the stimulus. Immunity may be transferred positively by antibodies or cells. Example, breast milk, vaccination, maternal immunoglobulin G, throat placenta, active norm to snake venom. May be positive immunity or active using antigen or attend nutrition organism toxide. This is the primary immune response. The development of accurate immunity began with a primary immune response and affect phases involving APC. APC called antigen presenting cells, trapping and capture of agents by disease. T cells transformation from a resting to an active state. An effector phase induction of other cells. For example, B cells or macrophage by active T cells secreting cytokines such as IL-2, IL-4, IL-5. This is also the secondary immune response. The primary immune response is the combined by the appearance of antigen-specific T cells such as T cells and CTL and immunoglobulin secreting B cells. The secondary immune response, once again, exposed to the same antigen, antigen-specific memory T and B cells are recruited much sooner and more efficiently. Immunoglobulin levels are consumably much higher. This is the T cells and the cell-mediated immunity. T cells develop from stem cells in bone marrow and mature in the thymus gland. After maturation, they migrate to lymphoid organs. The T cells are key cellular component of immunity. T cells have an antigen receptor to be recognized and react to the specific intracellular antigen via T cell receptors. T cell receptors only recognize antigen combined with major histocompatibility complex. It is called as MHC protein on the surface of the cell. MHC class contains two types. One is MHC class 1, then other one is MHC class 2. MHC class 1 is found on all nucleate cells. MHC class 2 found on phagocytosis, B cells and gastric cells. This is the overall structure of T cells only recognize antigen associated with MHC molecules on cell substrate. Okay, this is the clonal secretion. Increase number of effector T cells destroy the emulator. T cells regulate proliferation and activity of other cells of the immune system. B cells, macrophage, neutrophils, etc. defend against bacteria, virus, fungi, protozoa and etc. Cancer cell and transplanted tissue. Unlike humoral immunity, cell-mediated immunity is not transferred to the fetus. This is the T cell specific response diagram. Types of recognition by CD4 plus and CD8 plus. CD4 plus cells recognize antigens that have been taken of the antigen presenting cell called as APC, which present antigen fragment on the cell surface. CD8 plus cells recognize cells infected with virus altered cells such as tumor cells, which they then kill after recognition. This is the CD4 plus T lymphocytes. This cell is interact directly with other cells by releasing cytokines to control development of the immune response. Two types of T cells. THR1 cell active macrophage to destroy pathogens by phagocytosis and substance respiratory burst. Then other types CH2 cells help B cell to make antibody. This is the P lymphocyte. Specialized for the protection of immunoglobulin after differentiation into plasma cells, control pathogenic bacteria, prevent blood-brain infections, neutralize toxins produced by pathogens, 12% of total lymphocytes. This is also B lymphocytes can respond to peptide, carbohydrate and glycolipids. Usually require T cells can to respond to antigen but can also recognize antigen directly through surface immunoglobulin. TD and T1 antigen. B cells mature and proliferate in lymph nodes. This is antigen specific response structure. The classes of immunoglobulin. This immunoglobulin is otherwise called as Ig. This immunoglobulin contains 5 classes: immunoglobulin M, immunoglobulin A, immunoglobulin D, immunoglobulin G, immunoglobulin E. Structure and the biological of immunoglobulin. It contains four chain structure, 2 light chain and 2 heavy chain. Chains linked by bisulfite bonds. Chains are colliding in domains. 110 amino acids are present. In interchain disulfide bonds, this is the overall structure of immunoglobulin. It contains 2 light and heavy chain, then bond also etc. Terminal region of H and L chain. This is called heavy and light chains are the variable reagent. The variable reagent is the site where immunoglobulin combines with antigen. This reagent variability is responsible of range of antigen specificity. It is otherwise called as antibody diversity. The other domains are the constant reagents which are similar within an isotype of immunoglobulin. Using enzyme, a number of immunoglobulins fragments have been identified. Important ones are TRAB and FC. FC reagent is formed from heavy chains and determined isotype and so biological function. Range reagent also the effect of function. These functions are distinct from antigen binding, opsonization, complement activation, etc. This is the first type of immunoglobulin. Ig. Neutralize toxin. It is the activate component. It is the opsonization. It is able to cross placental barriers. 4 subclasses contain immunoglobulin G, immunoglobulin G1, immunoglobulin G2, and immunoglobulin G3 and immunoglobulin G4. Present in the main important function of immunoglobulin G is present in the highest concentration in blood. This is immunoglobulin A. Monometric and dimetric form. It is a dimetric form in secretory immunoglobulin A and is found in secretion such as tears, saliva, breast milk. Important roles of antiviral. It is the signature molecules of mucosal immunity. This is the other types of immunoglobulin IgM. It is the plantametric. It is a good agglutinator. It is a good and activating component. It is the first class of antibody to be synthesized by B cells after exposed to antigen. 10 antibodies binding sites, antigen binding sites. This is the overall structure of immunoglobulin structure. Then next we see immunoglobulin B. It is the active against parasitic infection, especially of helminths. Proton binds to receptor on breast cell base surface. Trigger mast cell denaturation. Expressed in high concentration in atopic individuals in response to alterations such as colon, animal cancer, etc. Immediate types in hypersensitivity reactions such as asthma. This is the HMC called as major histocompatibility complex. It contains types class 1, class 2, class 3. First of all, we see class 1. Class 1. Molecules expressed on all nucleated cells. Class 2. Molecules expressed on the small group of antigen presenting cells. That means APC. Class 2. Molecules may also be introduced to be expressed on other cells during immune reaction by interferon. This is the cytokinase. Cytokinase are short-range, multifunctional and short-acting mediator of cellular activity, especially within the immune system. Released stimulatively by activated T cells and other immune or non-immune cells. Act in a autocrine or paracrine manner. This is the classes of cytokinase. There are 4 major classes of cytokinase: interleukins, interferons, colony stimulating factor called as CSF, tumor necrosis factor called as TNF. It contains 2 alpha 1 type and beta other types. This is the natural killer cells. It is called as NK cells. NK cells, it is classed as part of the lymphoid system that have no differential surface marker. There are particularly affecting agents, various infected and tumor cells, also called range granular lymphocytes, Kilvia, gran enzyme and perforin. This is the antigen presenting cells called as APC. Normally, initiation of the acute immune response does not take place at the sites of injured or presentation of foreign organism. Antigen is take place by APC at the site of the inflammation and transferred to lymph nodes, where it is presented T and B cells. This is the also example for APC, antigen presenting cell. First of all, we see macrophages, B cell, dendritic cells. Dendritic cells is also called as DC. Macrophages and B cells recognize antigen through the immunoglobulin molecules. Dendritic cells produce and present antigen to native T cells via major histocompatibility complex class 2, most potent APC. This is the complement system. The complement system covers at least 9 plasma protein and some regulatory factor. They mediate several functions of the inflammatory process. Synthesized by macrophages or hepatocytes. Usually circulate at inactive proenzyme heat level. This is also complement pathway. This complement pathway gathered of sequential activation convert each proenzyme into the active site as inflammatory response. It contains 3 major pathways: classical pathway, alternative pathway, lectin pathway. Classical pathway bond immunoglobulin G and bond with immunoglobulin M. Alternative pathway attends antigen. This is final pathway is lectin pathway. This is function of complement system. Opsonization and chemotoxic immune tolerance. Clearance of acute inflammation, immune cytolysis, virus neutralization. Major complex causes the killing of the microbes through osmotic lysis. Thank you.